We clearly can identify and begin to identify a subset of individuals who are at much greater risk. The 1.7-fold increase in sudden death that I told you about is only for the entire population. If one goes to those at a much higher degree of risk, that risk turns out to be 5-fold or even greater. We've clearly learned a lot of novel biology of why arrhythmias may come about. And in many of these cases, not all, many of them now become drug targets, that is at least research on which ones may turn out to be superior drug targets. One of the things that's really become much more commonplace is just this view that we now do sequence-based biology. I think we talk about sometimes a pre-genome and a post-genome era. Since we are never going to get out of the post-genome era, a much better way of looking at our world is to say that we now firmly sit in an era in which we do sequence-based biology. And what's written here is in fact a set of ideals, if you will, or goals that I think 10 years ago would have seemed quite bombastic and impossible, but in fact it appears to be much more reachable than I think even the most optimistic genomicists really imagine, which is to know all of the human sequence. And when I mean all, when in fact the first reference human genome sequence was obtained, or let me put it another way, when the sequence of Craig Venter was first published and then Jim Watson's sequence was published, there was an additional 3% of DNA in one that was not obtained from the sequence of the first individuals. You've heard Dave talk about copy number variants. That means perhaps all of us carry a small bit of human sequence that might even be quite unique to us. So it's possible to know all of that sequence in the human genome and perhaps other genomes. We clearly now have the technology, and through the 1,000 Genomes and other projects, we will really know all of human genetic variation and its relationship to disease by the kinds of studies that I outlined. Not discussed here, but one of the things that we are making very rapid progress in projects such as the Encyclopedia of DNA Element Study or the ENCODE study is to know what all the functional elements in the genome really are. The number of other studies that have been done in cells in which one can in fact know all the signatures of cellular response. That is, if you alter a cell in a particular way, or if you expose it to a particular kind of chemical environment, what are all of its chemical or biological outputs. And Jeff pointed out a particular kind of synthetic biology, but long before we can construct entire genomes, that many of the same kinds of thinking and same kinds of experiments and same kinds of technologies are now possible to use in such a way that we can modulate at least all genes, at least all human genes in an experimental system, or perhaps even in some other animal model system. The difficult part that all of us are grappling with is when we have the sequence, when we have the variation, when we know all of this, can we predict function? And I think that's a big goal, but in many ways I think we're on the right path to begin to identify that problem much more specifically and solve it. And I think the technologies for most of these now exist, which is the kind of enthusiasm that you sense among all biologists and clearly those that study the human and study human disease. But there are many things we still don't know. We don't know the individual biology of all of the genes involved, and I think it's quite important for us to proceed in this direction. We still don't know the biological mechanisms that underlie sudden death. We may know more of the QT interval and how some arrhythmias arise, but we really don't understand it all, nowhere close to it. I think medically what's quite important is for us to understand what is it that triggers fatal arrhythmias. Exercise is one. We all know exercise is good for us. I don't want you to think otherwise, but there are many other kinds of physical or other activities that in fact can trigger. And there clearly are many other environmental factors that impact sudden death. Elevated glucose levels for one, however that comes about. And I'm quite sanguine that genomics will be a very large contributor to finding the answers to this as well. So let me stop here. I have my email if I can answer questions even beyond this room, but thank you for having me.